Hello, student. This is Dr. Faizan Mirza. This question regarding meiosis and how haploid diploid cells are forming. This came in October, November 2023, variant 42, and this question was a bit difficult. Many students found it a bit challenging. So let's discuss this question. And first of all, let's go through the facts which are told to us in this question. Yeasts are unicellular organism from kingdom fungi. This name is provided to us. We know the first name represents the genus and the second name represents the species. So the yeasts are unicellular organisms. Yeah. So this is genus. This is the species. So this is one species of yeast that can carry out either asexual reproduction by mitosis or sexual reproduction by meiosis. So if it can carry out both, so I've just underlined both facts here. Budding in this organism is a process where small daughter cell forms as a bud. So as a bud it forms. You need to just have the basic information of budding from your O levels. That's more than enough. But yes, a bud will always be similar to the parent that it is attached to, and it may be a bit different considering that the considering the size. But as far as the chromosomes are concerned, the variation must not be there because budding is an example of how the cells divide bisexually, like mitosis only. So as a bud on the parent cell, the bud contains a copy of a parent nucleus, which means it's a clone, and it's eventually separate from the parent cell to form a new cell. So this is an example of asexual reproduction. Furthermore, they are saying that this organism exists in two forms, haploid cells or diploid cells. Haploid cells contain one of the two different mating types, A and alpha. These are two mating types, so these must be haploid. So because only haploid ones can mate. Haploid cells already told to us. Haploid cells can only mate with other haploid cells of opposite mating type, which means A cannot mate with A, alpha cannot mate with alpha. So A and alpha can meet together. So keeping this in mind, let's move forward. Now let's move on to the figure 2.1, show the life cycle of this organism with its asexual and sexual reproductive stages. Keys provided to you. This circle within a circle, this shows to you A, mating type A. Circle within a circle, again, this is alpha. So let's move on. This one and this one, they both are cyclic events. So both are cyclic events 1 and 3. They are showing that an organ, the cell, which is A, it continues to be A, and a bud forms, and the bud then separates, and the bud then becomes an individual cell. So if it's a haploid cell, it stays haploid, it makes a haploid cell. Haploid cell is released, and the haploid cell continues its haploid life cycle. Example of budding, which is again similar to mitosis, some part of mitosis. So this actually is just a type of mitosis. Okay. Then the same thing is occurring with alpha. Again, cyclic stage one. Alpha produces a bud. The bud forms a separate nucleus. The bud separates. The bud is still this, or this cell is again a haploid cell, and the parent cell continues its haploid life cycle again. So we know they both are haploid. Then this A and this alpha, they are both haploid and they mate together. When they mate together, they form a zygote. The process is shown in stage two. Stage two is not a cyclic process. It's a stage that precedes this process where fertilization has occurred. So fertilization occurs, we see the cell which is diploid having both the nucleus A and alpha inside. So this must be a new variant. Why a new variant? Because it's having the genes or the chromosomes of A as well as the chromosomes and the genes of alpha. Then the same cell, which is diploid cell, it continues a similar event where again it produces, this is the diploid cell now, again, it continues mitosis and budding occurs. This produces a bud. The bud grows, the bud separates, and now the bud that separates will be a diploid bud because the cell already was a diploid cell. The diploid cell continues its life cycle in the same manner along the stage 3, which is again cyclic, and diploid cells, which are produced as a result of budding or again mitosis, they are removed and they actually are separated. Now, once this three occurs, then let's suppose if this organism is faced with lack of nutrients, in that case, this particular diploid organism, which is a new variant, it ends up producing spores. In spores, we know what we have studied in meiosis. We have studied in meiosis that a diploid cell ends up forming four haploid cells or a diploid nucleus ends up forming four haploid nucleus. And we know all four haploid nucleus, which are formed from a diploid nucleus, have different variations, are different variants because of different combination of alleles and chromosomes. So they are saying in ascus forms a sac containing four spores, and you can see each of these is having a haploid condition. Now, two are A, two are alpha, which means one diploid nucleus actually separated into four haploid nuclei, two of A and two of alpha. Germination occurs when conditions have improved. So when conditions have improved, something called stage 5 occurs. They have not mentioned what happens and which cells go there, but since from an ascus, the cell the arrow leads to is A, so we can say that the haploid cell A is moving along this line 5. And similarly, the alpha, which is again a haploid cell, is moving along the same line 5 here and combining, going with the cyclic event. So what we see here, we already have seen 1 and 3 are cyclic. We know 2 and 4 are in series. 3 and 4 are mandatory for 5 to occur. So let's move on. With reference to figure 2.1, state the number of stages 1 and 5 unto 5 that involve mitosis and meiosis. So just using the annotation from the figure, we figure it out that mitosis was in 1 and 3, which were both cyclic. 4 was the stage where meiosis occurred because a diploid cell formed 4 haploid cells. Produces new genetic variation. So genetic variation is produced, number one, when one meiosis occurred, and number two, when fertilization occurred. Similarly, showing only haploid cells. So 1 and 5 are the only parts where haploid cells were shown. One was the part where the cyclic events of A and alpha were occurring, and 5 when they were going and fertilize, they actually were moving from the spores and going to the asexual stage again. Shows the diploid cells. So diploid cells are there in 3 only. So why we use 3? Why didn't we use 2? Because 2 is not yet a diploid cell. This is a diploid cell. So 3, all cells are diploid. So other than that, there is no event where all cells are diploid. When there is a lack of nutrients, cells made in stage 3 will carry out to stage 4 to make spores, which will germinate only when conditions improve. So just explain how this type of reproduction make the spores during stage 4 as advantageous for this organism in the changing environment. So we know the spores which are produced during stage 4 are all different from one another because they are all haploid nuclei and they arise from diploid cells. Stages of meiosis, such as crossing over between non-sister chromatids of homologous pair, followed by independent assortment of chromosomes, and therefore alleles, some fact that we learn in meiosis, they produce different combinations of alleles. Better variants are produced, and this also leads to random fertilization of haploid gametes later, resulting in much fit variants better adapted for changing environment. Haploid and diploid cells of this can carry out asexual reproduction, suggests why new harmful recessive mutation may not be having a damaging effect on asexually reproducing population of haploid cells or an asexually reproducing population of a diploid cell. So why haploid cell and diploid cell may not be affected? So you have to mention them separately. All haploid cell will not be exact clones. We know that. So only those haploid cells will be affected that have the allele with recessive mutation. Other cells of haploid will not be affected. Haploid cell which will be having the recessive mutation won't survive and will be removed from the population. In diploid cells, although, since there is a pair of chromosomes, so the chances of a dominant allele which is normal will be there together with the recessive allele which is mutated. The mutation will not be expressed in the phenotype in the presence of the dominant allele. Lastly, the state 2 features other than reproduction using spores for the kingdom of fungi. So we can explain, we can just state that kingdom fungi of organisms which are eukaryotes. Organisms of kingdom fungi have chitin in the cell wall. They are always heterotrophic nutrition, release extracellular enzymes in the food to absorb solvent products. So I hope this video was helpful for you. I would recommend that you go back and try to attempt this question 2 of this particular paper, and see if this video is helpful for you. That's it from my side. Thank you so much.